start. My name is Ashley George, and I'm a postdoc in Nadia Roan's lab at the Gladstone Institute. And it is my great pleasure to introduce today's Infectious Diseases and Human Health seminar speaker, Dr. Brad Jones, who is coming to us virtually from Weill Cornell Medicine. Dr. Jones earned his Bachelor of Science from Simon Fraser University and his PhD from the University of Toronto. After that, he completed his postdoctoral training at the Ragon Institute, Massachusetts General Hospital, MIT, and Harvard. Dr. Jones then joined the faculty of George Washington University before moving to Weill Cornell Medicine, where he is currently an associate professor of immunology and medicine. Dr. Jones's research focuses on understanding the relationship between HIV and the human immune system, including ways cellular immune responses can be harnessed for new HIV cure strategies. Importantly, his work spans basic science through to preclinical models of HIV infection and through to clinical trials. Additionally, his lab studies why shock and kill HIV cure approaches have been effective in model systems, but less so in clinical trials, by understanding what factors may underlie differential susceptibilities of HIV infected cells by killing by cytotoxic T cells. Today, Dr. Jones will be speaking on silence and resistance and HIV reservoir persistence. If you have any questions, please wait until the end of Dr. Jones's presentation. And at that time, you may either unmute your mic or type in your question in the chat. And with that, please join me in welcoming Dr. Jones.
Great, thanks so much, Ashley. I'll just share my screen. Okay, thanks so much, Melanie and Nevin and all for the invitation. Really a pleasure to have this chance to present to you today. And I actually wanted to start with a slide that actually takes my background a little bit further, because my first start in a biology lab was actually in Nevin's lab a little while ago now, in 1999. Here I am, so really grateful to Nevin for kind of giving my career this great kickstart. So fast forward a bit, I wanted to introduce our Delaney grant, because I know, of course, the Hope Delaney is headquartered at the Gladstone Institute. So we have a Delaney called Research Enterprise to Advance a Cure for HIV, or REACH. And just a little bit of a who's who before jumping into some of our aims, and then I'll show you how our work is contributing to those broader aims. So I lead this Delaney along with Marina Kasky, and our executive committee is Sabrina Brumet, Michelle Nussenzweig, Paul Benash, Damli Nakanjako, as well as Doug Nixon, and Jessica Salzwiedel is leading our community engagement. So in our Delaney, we are exploring the hypothesis that cellular immune responses, NK and T cells, can be harnessed to achieve durable remission and eradication of HIV reservoirs. And some of what we think are kind of our unique angles here are in remission, this idea that there's limited mechanistic understanding of cases of partial success. So when people have received bNAb infusions, there's some evidence that there's CD8-mediated control, but that's not understood particularly well. There's a lack of effective strategies to limit immune escape, and there hasn't been that much testing of combinations of humoral and cell-mediated immunity. More on point for what I'm going to be talking about today in terms of eradication. We feel there's been perhaps a bit of an oversimplification of the relationship between CD8 and NK cells and the HIV reservoir on ART. This idea that the HIV reservoir is not visible to the immune system on therapy is something I think there's some evidence against, which I'm going to present today. And I would put forward this idea that there might be a lack of appreciation and strategies for overcoming this aspect of the reservoir, where cells that harbor intact proviruses appear to have been selected for being resistant to killing by these immune effectors. So today, my talk is going to focus squarely on these two aspects of the overall program. So to take a step back, we know that CD8 T cell responses play a critical role in controlling HIV replication. In this cartoon, you can see the first line of evidence supporting this, which is in primary infection. Once you have the emergence of the HIV-specific CD8 response, This coincides temporarily with when viral load is coming from the peak down to a set point. And there's a number of other pillars of evidence for this really well-established role for CD8 cells. Another one being the fact that there's strong genetic associations between having particular HLA-B alleles and control of HIV replication. We can also see this in vitro and in mouse models. So I want to give a little bit of a preview of a model that I'm going to be talking about a bit later in the presentation, which we call the participant-derived xenograft mouse model. And in these immunodeficient mice, what we can do is engraft them with CD4 cells from people living with HIV, in this case from an elite controller, infect the mice with HIV, and then simply either give autologous CD8 cells or no CD8 T cells. And to the point that CD8 T cells play an important role in controlling viral replication, you can see that just the addition of CD8 T cells is sufficient to drive up to a 4-log reduction in viral load in this mouse model. So there's a lot of evidence that CD8 cells have a lot of potential to exert antiviral activity. And so the question is, once we suppress antiretroviral therapy, suppress HIV with antiretroviral therapy, why is it that this arm of the immune system can't essentially finish the job and eliminate the reservoir? And we know that this, of course, doesn't happen because if you stop antiretroviral therapy, the virus rebounds, usually within weeks in most people. So the classic model of why CD8 T cells can't eliminate this reservoir, it really centers around viral latency. This idea that if a cell is latently infected, it's not going to express any viral antigens. It's not going to be seen by CD8 T cells, and therefore it will persist. And on the other hand, if a cell is expressing HIV, it will die either by viral cytopathic effects or by cytotoxic T cell responses. And of course, this issue of latency is absolutely critical and paramount. But what I'm going to show data to you today in support of is this idea that there are also some productively infected cells that don't seem to be getting killed by CTLs to the degree that we think that they should. So is there something else going on with why these cells are able to apparently resist being killed by cytotoxic T cells? And so moving beyond this model, here's a kind of modern conception of the various parameters that are at play, the various dimensions in HIV persistence. This is from a Nature Medicine review article. And so there's the dimension of different tissues harboring the reservoir, so an anatomical dimension. There's also the fact that different types of cells can be infected with HIV and harbor a reservoir. So a lot of focus on lymphocytes, but we know that there's also myeloid cells that can be infected and contribute to this. And now I think moving a bit beyond this dichotomy of latent versus activated, there's a growing appreciation that infected cells can exist on this spectrum of latency. Some can be in very deep latency. They may be blocked and locked, which I know is a very key topic of study at Hope and the Gladstone Institute. Or they might be fully activated, or they might be somewhere in between on this spectrum. So for these cells that are not silent, the cells that are expressing HIV, again, there's this idea that one way they might be able to persist is by being able to resist killing, even when they're recognized by a cytotoxic T cell clone. And so I'm going to focus on this for the first data part of the talk. And then if there's some time, I'm going to get into some data from our mouse model that shows what CD8 selection looks like at the level of the genomic integration landscape. Okay, so where did this idea that some cells are resistant to immune killing come from? And I'll credit this largely in part to work from Kira Clayton, who is in Bruce Walker's lab, who studied HIV-infected macrophages first and showed that macrophages are really hard for cytotoxic T cells to kill, even when they're good targets. So here's an experiment where Kira took CD4 T cells, and you have peptide pulsed or unpulsed cells, and you mix them with increasing effector target ratios of a cytotoxic T cell effector. And you can see nice killing of the peptide pulse cells. In parallel, though, here's macrophages, which are exposed to the same conditions, and you can appreciate that they're not succumbing to killing induced by the cytotoxic T cell. This is not because they're not being recognized. You can look at the CD8 T cells in both cases, and you can see that the cells are degranulating, whether they're cultured with infected CD4 cells or infected macrophages. So they're, firing their shots against these target cells in both cases, but you can appreciate that there's a major difference in the degree to which this results in death of the target cells. And Cure subsequently followed up on this and showed that macrophages are also resistant to killing by natural killer cells. So two different types of effectors, even though they're somewhat related, and the same observation that macrophages are hard to kill. So this essentially got us thinking along the lines of, okay, different cell types have different susceptibility to being killed, but what if we zoom in a bit closer and ask within lymphocytes themselves, within CD4 T cells, are there some cells that are harder to kill by cytotoxic T cells than others? The other line of evidence that got us thinking in this direction were recent publications from Matthias Lichterfeld's group and others, which shows that the persistent HIV reservoir in people is perhaps not quite as silent as we might have thought. And here's some longitudinal data from clinical samples looking at cells with intact HIV proviruses that are transcribing HIV RNA. And what you can appreciate is that even after a year of art, even after 9 years of art, there continue to be an appreciable proportion of these that are expressing HIV RNA, and a substantial proportion of these that are also expressing high levels of HIV RNA. So again, this question of why are these cells not being killed by CD8 T cells, and particularly in light of some evidence from us and others, which suggests that HIV-specific CD8 T cells are continuing to see these cells on art. I'm actually going to skip over this. So in 2018, we put forward this hypothesis that latent HIV reservoirs exhibit resistance to elimination by CD8 T cells for the reasons that I've essentially gone through. So what do we know about the ability of target cells to resist killing? And I think this is something which, other than Cure's work with macrophages, is still fairly new in the setting of HIV, but it's something that's known, and a lot of the mechanisms are known in the setting of immuno-oncology. And so I'm going to show figures from two different review papers here, because I haven't found a single review paper that shows all of the mechanisms of CTL resistance in a target cell. But here's the first one, and these are all events that happen downstream of a target cell receiving an inducement to die from a cytotoxic T cell. So either perforin or granzyme, or a death receptor, so Fas ligand or trail. So we often think that if a cell receives these signals, it's doomed to die. But the target cell has a lot to actually say about this by expressing factors that can be inhibitors of these inducements to die. So one example is that granzyme B can act by cleaving BID into truncated BID, which can then form pores in the mitochondria and induce mitochondrial cell death. But pro-survival BCL2 family members, such as BCL2, can counteract this by sequestering the truncated BID and stop it from forming pores. Another mechanism of cell death acts through caspase cascade, so caspase 3 and 7. But the XIAPs, so BERK3 and BERK5, are able to directly antagonize caspase 9 and caspase 3 and 7. So a cell that overexpresses XIAPs will be less likely to die. The final thing that I'll point out on this slide is CFLIP, which is an antagonist of caspase 8. So if a cell receives an inducement to die through a death receptor, overexpression of CFLIP can allow it to, in some cases, resist dying. This is the second slide, which shows us another series of mechanisms. This one's much more biophysical in some ways. So if a cell has a highly ordered lipid membrane, perforin is less able to form pores in it. Similarly, high exposure phosphatidylserine inhibits perforin pore formation. Really interestingly, it's been shown that cells that are less stiff are harder to kill. You can think of it essentially as a balloon. If it's fully inflated and you punch a hole in it, like perforin does, that's going to result in the balloon popping. Whereas if you have a partially deflated balloon and you punch a hole, you might have more time for, in this case, the cell to repair itself from these pore formations. Also, granzyme B inhibitors like serpin B9, the release of proteases into the synapse, which can cause degradation of perforin, and others. So there's a lot to go through, and I'm going to transition back to HIV, but I'll end this connection with immuno-oncology by referring people to this really beautiful review paper from the Valitutti lab, which summarizes rapid, slow, and constitutive ways that different types of cancer cells can defend themselves against cytotoxic T cell attack. So, sorry. Okay, so with this background, we sought to address two knowledge gaps that we think are potentially important for HIV persistence. One is, are some T cell subsets intrinsically harder to kill than others? For example, are central memory cells harder to kill than effector memory cells? The second knowledge gap is, if we zoom in to a particular T cell subset, such as central memory cells, is there heterogeneity in susceptibility to CTL killing? So we're approaching this problem from the two extremes at once. In the first part, really tackling the fact that CD4 T cells come in all kinds of different flavors and differentiation states. And the second is zooming in closely on one type of CD4 T cell and looking at mechanisms of resistance. So the second part is much more advanced than the first part, so I'm going to spend a lot more time on it. But I'm going to show just a few slides showing our efforts to tackle this issue of T cell populations and diversity. So two of the ways we can think of T cell heterogeneity are shown here. First of all, naive cells, when they mature, can be polarized to a number of different types of subsets, so Th1, Th2, Tregs, etc. And then furthermore, essentially within each of these polarization tracks, you have different phases of T cell maturation. So naive cells converting into stem cell memory, central memory, effector memory, and so on. So to get a feel for this diversity, what we've decided to use is a site-seq panel. So this is single cell RNA sequencing, and I think people will, I hope, be somewhat familiar with this. We have a panel of antibodies which have, instead of a fluorophore, a DNA oligo as their tag. So in the end, when we do our single cell RNA sequencing, we get information not only on the host transcripts, but the level of antibodies that bound, which inform us on the expression of different transmembrane proteins. So we begin to apply this to CD4 cells. We pulse with a peptide. We subject to killing by a cytotoxic T cell clone. And you have non-peptide pulse cells in the same well. So the non-peptide pulse cells we're calling bystanders, and the peptide pulse we're calling survivors. And the first thing that stands out is you can see that within the survivors, you have this striking overrepresentation of naive cells and this depletion of effector cells. And you can see the overlay here with some of the protein expression as well as mRNA expression profiles that inform us the identity of these different clusters. So as we move along the cell maturation pathway, cells become more and more susceptible to being killed. And again, these are pulsed with peptides. So this is not telling us about antigen presentation. This is really about the intrinsic susceptibility of that cell to succumb to a cytotoxic T cell hit. We, of course, have a lot of depth that we can go into with this single cell RNA-seq. So I'm going to show just a few things that we've observed so far. Here are the cells that survived in blue, and these are the bystanders. And what we can see, which we reported previously, is that there's overexpression of this pro-survival factor BCL2 amongst the cells that survive. But the single cell data can also give us a look into the heterogeneity of this. So it's not, of course, that all survivors are BCL2 high. There's a population that's BCL2 high, which may have survived through this mechanism. And there's other cells that have survived, presumably through other mechanisms. We also see overexpression of BOC2 in the cells that survive killing. We're kind of just at the beginning stages of exploring this. And another hit that caught our eye is NEAT1, and NEAT1 induces autophagy. And this is one way that a cell can recover from a cytotoxic T cell hit, because granzymes can damage organelles, which can lead to cell death. But if you recycle, you remove the organelles by autophagy effectively enough, the cell can potentially recover. So we have found this BCL2 hit many times in many different modalities. And this is one mechanism of resistance that we've really taken all the way through to validation, showing that ex vivo cells that harbor the HIV reservoir tend to be high for BCL2, though there's some heterogeneity. And if we add a BCL2 antagonist, it enables the CTL-mediated clearance of HIV reservoir cells in vitro. So those are just some initial findings from this first data set, which, as I mentioned, we're much earlier on in terms of developing or analyzing all the data that comes from this site-seq. But we see that naive T cells are very hard to kill. They can also harbor intact proviruses, others have shown. So this may be a kind of sanctuary for intact proviruses. We're exploring mechanisms for why, but one thing we note is that naive cells don't express any FAS, so they're not going to be subject to FAS ligand-mediated killing. But there are, of course, other resistant populations that exist in our data set, and we're in the process of doing more in-depth characterization of these. The second part, though, on which I want to spend a lot more time, in which our data is much more developed, really focuses on this central memory population. So we decided to focus on this population, both because it's a critical long-lived HIV reservoir, and also because it has this kind of intermediate susceptibility to being killed by CTL. It's not as resistant as naive cells, but there are some cells that survive killing, even when they're pulsed with peptide. So can we discern what might be different about these cells? By moving to these central memory cells, we can also transition from this peptide pulse model to a more physiologically relevant, this HIV infection model. Sorry about this. So we, one of the first hits that we found in these experiments before we moved to a site-seq panel is overexpression of this gene called EZH2. And we can see from these RNA-seq data that cells that were exposed to CTL and were also expressing HIV GAG have the highest level of expression of EZH2. And want to mention briefly that this has been followed up on by an alumnus from the Gladstone Institute, Andrea Grammatica, who had a nice poster at the Miami meeting where he showed that this licensed drug, tazemetostat, which is an EZH2 inhibitor, can increase MHC1 antigen presentation and enhance killing by cytotoxic T cells. And this is just a bit of data from those experiments showing a greater proportion of killing of infected cells after exposure to tazemetostat. So those were some early results, but what we've moved on to now is kind of a more refined method that we are using to build a foundation for understanding CTL resistance in this population. How we generate our target cells is shown here. We began with naive CD4 cells, and we activate them under non-polarizing conditions to make these non-polarized central memory cells. Again, the goal being to make a population of cells that is relatively homogeneous, which we think is going to help us drill down on some of the individual host factors that might make a cell resistant to killing. And we infect with JRCSF, and these are ready then for the killing assay. The trick that we start introducing here to have a really precise control is to use a pair of viruses that are wild type or have a point mutation in the CTL epitope that's being targeted by the clone that we're going to use for these killing assays. So the reason this is important is it allows us to label these two populations of cells, mix them together in the same well so that they're present in the same environment. Both populations are infected with HIV, but the only difference is that one of these populations is subject to killing and selection by the CTL clone, whereas the others are bystanders that are present in this same well. So at the end of this experiment, we have a simple two-way comparison between the escape mutant viruses that we call the bystanders and the infected cells that survived killing. We then subject these to bulk RNA sequencing, as well as single-cell sequencing, RNA-seq, site-seq, as well as TCR profiling. So before, of course, spending a lot of money on these site-seq assays, we wanted to do a bit of a sanity check and ask if whether in this in vitro system that we devised, are the cells that survive a single round of killing actually resistant to being killed? And to test this, we designed what we call a sequential killing assay. So this is the first round of killing that is analogous to what I just showed you. There's a pair of wild type and escape mutant viruses. And then the trick is, after this first round of killing, we come in with a second CTL clone that recognizes a different epitope, which is present in both of these two virus strains. So if there's no such thing as CTL resistance, in the first round, you'll kill off the wild type cells. And then in the second round, you'll kill off both the wild type and escape cells to a similar degree. If there is CTL resistance, then you'll see that the cells that survive the first round preferentially also survive this second round of killing. And so we've done this experiment many times, and I'm just showing some representative data here. Here's a first round of killing just to show that the SL9 specific clone does induce killing of both populations. But this part of the graph is what's really critical to the point. So here's a first round of killing where we see that the wild type cells are killed very efficiently by the CTL clone, whereas the escape mutant cells are not killed. And then when we come with a second round of killing with an SL9 specific clone, what we see is that we nicely kill off the cells that were not selected in the first round, but there's no further reduction in this small population of cells that survived this first round of killing. So there's a basement, really. There's this core rare population of cells that is able to survive through both rounds of killing. We've done just a little bit of microscopy. Here's an infected cell in green of cells taken out from these survivor wells. And you can see this interacting with a cytotoxic T cell clone and not succumbing to killing. And I think I saw Rob further on the call, so shout out to him for help with these microscopy experiments. All right, so we think it's going to be important to study this population. So let's move ahead with performing the omics on these cells relative to the bystanders that were in the same wells. So I want to show what the raw data look like here, because I want to make one point really clearly, which is that for both the survivors and the bystanders, we're gating on cells that express HIV. So here we're using two antibodies because we want to be sure that we capture HIV expressing cells. When we haven't added cytotoxic T cells, we can see that there's nice populations of infected cells in both conditions. Where we've added our CTL clones, we've killed off many of the wild type infected cells, but as expected, the escape mutant cells survive. And in both cases, we're comparing HIV expressing cells to other cells that are also expressing HIV. So this is not about latency. This is about cells that survive despite expression. And this is the bulk RNA-seq data. And with the principal component analysis over here, what you can see is as expected, where we haven't applied CTL pressure, the cells infected with the wild type or the escape mutant HIV cluster together with each other. These are all biological replicates from different donors. When we add cytotoxic T cell clones, as you can imagine, we have perturbation of the transcriptional signatures of all cells in the well because the cytotoxic T cell clones are making inflammatory cytokines and such. But what's really important is this difference here. So the cells that were targeted by cytotoxic T cells and survived cluster separately from the cells that were also HIV infected, present in the same well, but not targeted by CTL clones. This heat map shows the same data set, and again, reproducible across different days and different donors, and quite a lot of differential expressed genes between these two conditions. So there's a real signature of these survivors. Our challenge now is to kind of break this down into bite-sized pieces and digest what the mechanisms are that, that underlie this. So if we do go-term analysis for biological processes or cellular components of this difference between the bystanders and survivors, these are the main processes and components that are implicated. So interferon signaling comes up as being high in cells that survived killing by CTL relative to bystanders that were in the same well. We have some ideas for why that might be that I can talk about in the question part, but no validation just yet. The second component that we find very interesting is cytoskeleton and junction formation. So there's underexpression of genes that code for a lot of adhesion molecules, and as well as of genes that regulate cytoskeleton formation. And we think that two things are going on here. So as I mentioned previously, the physical stiffness of a cell determines how susceptible it is to being killed by cytotoxic T cells. That's been shown in melanoma cells quite extensively. And also, if a target cell lacks the ability to form junctions with a cytotoxic T cell, you can imagine how that would prevent it from effectively being targeted and killed. So this appears in both of these analyses. And then we also see implication of ribosome and translation, where these ribosomal components tend to be overexpressed in the cells that survive killing. This is again something that we don't understand at this point. So if people have any suggestions for this in the questions, those would be gratefully received. So taking a bit of a closer look at these pathways with KEGG analysis, these are again pathways involving formation of cell-to-cell junction, suggesting that the target cells that survive may be less able to form stable contacts with cytotoxic T cells, as well as regulation of actin cytoskeleton translation pathways and others that I'll talk about a bit more. Throughout this, there's interesting potential connections with immuno-oncology field. So, so there's less cholesterol homeostasis here, and it's been shown that if you actually stiffen different types of cancer cells by depleting cholesterol, it can enhance their susceptibility to being killed by cytotoxic T cells, both in vitro and actually also in an in vivo model. We have also the single cell data that corresponds to this bulk RNA-seq data. And what you can see here as well is that the cells that survive killing cluster quite differently from the infected bystanders that are present in the same wells. We again see some of the genes that we have consistently seen in exploring this phenomenon of resistance. So overexpression of BCL2 that we and others have reported as conferring resistance to cytotoxic T cell killing. And tantalizingly, we also see at the protein level overexpression of PVR. So PVR is a ligand for TIGIT. It's highly overexpressed in the cells that survive killing, or highly statistically significant, I should say. And there appears to be an interesting convergence here with a recent paper from Matthias Lichterfeld's group, where they were able to phenotype ex vivo cells that harbor the HIV reservoir. And one of the genes that really stood out as being overexpressed in cells with intact proviruses was PVR. So one thing we want to continue to do, and one thing that there may be opportunities for collaboration with folks on this call, is take these data sets that we're generating on the profiles of cells that are resistant to killing, and then cross-reference those against characterization of real reservoir harboring cells from people to ask which putative resistance mechanisms might be at play. So along the lines of this observation. I know there's a lot of data here, but just to briefly show a few things, these are all protein expression data. I mentioned earlier that there was underexpression, as indicated in red here, of a number of different adhesion molecules. So beta-2 integrin, ICAM, yeah, those are the ones. And also of underexpression of MHC1, as you might expect in the cells that survive killing. And if we look across the different clusters that are defined by the 10x data, we generally see that this is consistent across many different clusters. So across many different types of cells within this population, cells that underexpress beta-2 integrin, for example, preferentially survived this killing. So there's a lot of leads to follow up on, and I'm showing a lot of unpublished data, so we don't have a full validation for all of this yet, but I'll tell you kind of the directions that we're working in and the data that we have so far. The first area is cytoskeleton and cell stiffness. And I mentioned this study earlier from Morgan Hughes's lab, which emphasized that in order for a cytotoxic T cell to be able to kill, it has to be able to exert mechanical force across the synapse on the target cell. It has to essentially stretch the target cell for perforin to be able to perform its function properly. And if the target cell is less stiff, this is a less effective process. So do we have any evidence that cells that survive killing in our assays are actually less stiff than the bystanders? To test this, we've teamed up with Scott Manalis at MIT, and he has this instrument that is quite similar to a flow cytometer conceptually, but rather than measuring fluorescence, it applies acoustic fields and is able to measure the deformability of cells as they pass through this sensor. And so what we can see is that even though there is appreciable overlap between the two populations, the cells that survive killing, shown in blue here, tend to be significantly less stiff, so they're more deformable than the cells that were bystanders, also HIV infected in these same wells. So some initial validation of this idea, and now we're experimenting on ways to stiffen up these target cells and see if that improves their susceptibility to killing. So, so the question basically with cell stiffness is the HIV reservoir squishy, and how to measure? And one of the ways to do this might be to stiffen the cells and see if this improves susceptibility to killing. The second big lead that we have here is cell adhesion. So are cells that survive killing and potentially cells that form the HIV reservoir also slippery, essentially? Are they hard for the cytotoxic T cells to form synapses with? So we're now able to perform adherence assays to ask if survivors bind less well to things like ICAM1. And this is something that we also believe that we can apply to bona fide HIV reservoirs, ex vivo CD4 cells from people on ART, and the hypothesis being that cells that don't adhere to the matrix will be enriched in HIV DNA. The third lead that we're following up on is the question of whether metabolism plays a role. Are metabolically quiescent cells resistant to killing, and are HIV reservoir cells relatively quiescent? So there's an important link between metabolism and susceptibility to CTL killing, in that the susceptibility of cells to death by granzyme A and granzyme B is dependent upon reactive oxygen species in the target cell. And this was shown by Judy Lieberman's group, who showed that adding scavengers to of these superoxides is actually effective in blocking killing by cytotoxic T cells. So these are generated as a result of various metabolic pathways and may explain why glycolysis, glucose metabolism, as well as hypoxia. These are all significantly down in cells that resisted or were able to survive killing by cytotoxic T cells. We can also look at the genes in these pathways at an individual level from the single cell RNA-seq data. And what we can see is all of these genes being down in survivors, shown in blue, and these genes occupy these very central roles in this metabolic pathway of glycolysis. So knowledge gap number 2 here, within a given T cell subset, is there heterogeneity in susceptibility to CTL killing? And the main message that the main take-home message is the answer, I believe, is yes. We can show that functionally. Cells that survive 1 round of killing are less susceptible to a 2nd round of killing. And we can see that in the transcriptomic and site-seq data, where there's really quite a distinctive profile of cells that survive killing relative to bystanders that are closely matched in terms of environment. We are pretty advanced in terms of validating and showing that some host factors can enable CTL resistance and can be overcome. We've published on BCL2, and I mentioned Andrea's story with EZH2 as a factor that suppresses MHC1 expression and prevents killing by CTL. And then 2 leads that are still in the process of being validated, but I think are supported quite nicely by the omics data, is that there's underexpression of adhesion molecules in cells that survive killing. So are these cells, as I mentioned, slippery, and cytoskeletal regulation, as well as regulation of some lipids, is also different in cells that survive killing. So are there differences in the stiffness? In the last few minutes, I wanted to squeeze in just something else because I think it's important to block and lock and maybe of interest to the HOPE Collaboratory. So I do want to leave some time for questions. So I'm going to go quickly, but I hope people will be aware of recent studies which have made the case that CD8 T cells may be selecting for HIV proviruses in transcriptionally disfavorable regions of the genome, and particularly in elite controllers. So what we've been doing is seeing if we can apply an in vivo model which adds CTL pressure or doesn't have CTL pressure to see if these features can be recapitulated, and if so, to leverage this model to further define the footprint of CTL selection on the integration site landscape. So I only have 2 slides on this, and it's also going to be presented at CROI if you're interested in seeing more. But essentially, the mice are engrafted with CD4 cells infected with HIV and either given CD8 T cells or not given CD8 T cells, and we can follow them out to 93 days. Viral load is suppressed, again, up to 4 log over the course of this experiment. And this is the immunodominant HIV-specific response to KK10, which we can see it expands and is retained at very high levels in these mice. So there's quite sustained CD8 T cell pressure. The proportion of cells with HIV DNA is lower amongst these mice. Surprisingly to us, we don't see skewing in terms of the percent of intact proviruses, but we do see selection for latent proviruses, where the fraction of intact proviruses that are expressing GAG is much lower in the cells that have been through selection. So what we see amongst the integration site landscape between these 2 groups of mice is that if you look at the no CD8 mice, what we see in gray here are all the integration sites that are unique, that were only found once. So most infected cells are not clonally expanded in these no CD8 mice. In contrast, where we've added our CD8 T cells, we see that most of what's survived and is left are these few clones of expanded cells that seem to be able to survive and thrive despite this CD8 pressure. What we've seen when we've taken a closer look at these proviruses are a few things. The proviruses in the CD8 selected mice are more statistically significantly more likely to be outside of genes, as well as further from the nearest gene than in mice that did not have CD8 T cell pressure. They're also more likely to be in the opposite direction of the nearest host gene, as well as more likely to be in the periphery of the nucleus, which is a less transcriptionally active environment. So this is, again, work that's in progress, but I wanted to mention it again because perhaps this is a model and some room for collaboration with the HOPE Collaboratory, a model perhaps for some block and locked proviruses that are selected by CD8 T cells. So just to put this all together in my last slide, I've talked about HIV resistance to cytotoxic T cell clones, and then a little bit at the end on HIV latency and silence. So how do we think of these two factors that might enable resistance? And when I talk about resistance to killing, I often hear people say, like, oh, great, even if we overcome latency, we have to deal with resistance to killing. But what I would encourage people to do is think about it more on this type of spectrum. So we know there's active reservoirs, cells that are actively transcribing HIV that persist, that are detectable in people on long-term therapy. So maybe we can make a dent in this part of the reservoir by targeting and overcoming mechanisms of resistance, such as BCL2 overexpression. To cut a bit deeper into the reservoir, we probably have to combine with latency reversing agents. And then at this end of the spectrum, there may be these block and lock proviruses in deep latency that perhaps we don't have to worry about at all because they may never reactivate. So with that, I hope I've left some time for questions. Yes, and I'd like to thank Louise, Alberto, and Emmy, who have been the PhD students leading this project, as well as Paul Zumbo, who's performed bioinformatics. Dennis Cupertino's done outstanding work on this project as well, and I know is applying to the UCSF PhD program. Highly recommend him. I want to thank also our collaborators, Kira Clayton, I mentioned her work at the beginning, and our clinical collaborators who provide the samples. And thanks as well, of course, to all the study participants who donated samples for the work that I presented. With that, very happy to take any questions or comments.
Maybe I'll start with a question. Thank you for that amazing talk, Brad. That was really, really impressive. So I have a couple of questions. The first one regarding the first part and the second one regarding the second part. So in the very beginning of the talk, you presented the data with the macrophages versus T cells, where the macrophages are most resistant and the T cells are much more susceptible. But then you later showed that amongst the T cells, you also kind of have a range. So is the idea that there's kind of a whole spectrum of susceptibilities, and CD4s is kind of in the middle? And related to that, the top of the CD4 spectrum was naive T cells. Is there any evidence that naive T cells that harboring latently infected cells, that it increases over time, which you might expect based on the data that you present?
Yeah, so thanks very much, Nadia, for the question. And yeah, the way that I would frame it is, if you look at completely different cell types, like macrophages to lymphocytes to, if we get into cancer as well, melanoma cells, there's going to be intrinsic differences in susceptibility to being killed. But CD4 cells, at least because they're so diverse, will also have different subsets that are differentially susceptible to being killed. Like you said, naive cells are particularly hard to kill. As well as, zooming in a bit closer, even kind of cell-to-cell differences within a given subset that might determine this outcome. I'm sorry, I forgot the second part of the question.
Naive cells that are infected, is there any evidence that it increases over time, like over decades of art? Yeah,
that's a really good question. And yeah, the hypothesis I agree with. So this idea that if naive cells are a sanctuary for intact proviruses, if you track people over time, do you see that essentially the ratio of intact proviruses in naive to memory cells gets skewed? I'm not aware of any studies that have looked at that, but I agree it would be an interesting hypothesis to explore.
And then the macrophage resistance, is that because I like your balloon analogy for the stiffness thing? Is it because they're like very unstiff, if you will. I mean, at least I imagine them to be much less stiff than T cells. Do you think that's a big part of it, or you think it's more complicated than that?
Yeah, even though we have this very clear resistance profile, I think the mechanism is still not that well understood. I mean, if you look at this in microscopy, you have a real David and Goliath situation happening with little cytotoxic T cells trying to tackle a large macrophage. One factor that is overexpressed in macrophages, which I mentioned briefly in the talk, was SERPINB9, which is an inhibitor of granzyme B. So I would propose that SERPINB9 may play a key role in this, but the experiments of going in and knocking down SERPINB9 and seeing if that makes the cells more susceptible to being killed haven't been done yet. So we're not able to clearly say that. But antigen-presenting cells, in general, it could make sense why they might want to resist being killed when they encounter T cells. So that would be kind of the conceptual reason for it, I think. But even looking at those differences between cell types, I think the mechanistic underpinnings are not too well known.
My last question relates to the second part of your talk, when you said that interferon signaling is increasing the survivors, and you said that you could speculate a bit on that. So can you go ahead and speculate a bit on that? I'm curious what you think those data mean.
Yeah, thanks. So I think it has to do with teasing apart cause and effect here. So we've tried to really precisely control for the environment of the cells by having wild type and escape mutant viruses. But there's kind of a higher resolution environment that you can think of, which is that cytotoxic T cells are going to form closer contacts with the cells that go on to survive. And what Cure showed with macrophages is when those T cells fail to kill their target, they produce a lot of interferon gamma, a lot of cytokines. So those kind of intimate contacts where the CD4 cell essentially lives to tell about that close encounter might explain that result. We might be seeing those signals as a result of surviving rather than a cause of surviving. So we're doing experiments right now as well, and asking the question, if we block all de novo translation at the time that we do these CTL killing assays, do we continue to see this resistance profile? And I'd also be interested in applying a method like your PP slide to kind of trace back these cells that survive killing and ask the question of what they look like potentially before they had these close encounters with cytotoxic T cells.
That's really interesting. Thank you.
Thank you.
We have a question in the chat from Fran. Are you planning on measuring cholesterol levels on the surviving cells, particularly plasma membrane cholesterol?
Yeah, so it's a really good question. So we've so far been focusing on transcriptomics and protein expression with CytSeq. I think it would be great to do some lipidomics as well. We haven't taken any material steps towards doing that so far. And if there's folks on the call who have those expertise who might be interested in collaborating, we'd be really interested in talking to you about it. But so far, no direct measures of the lipids.
Gus, do you have a question? You can go ahead and unmute yourself.
Yes, I have a question. Thank you, Brad, for this wonderful talk. My question is related to, I would expect that the CTLs, that the cells that are attacked by the CTLs and actually died, maybe share some of the changes in gene expression that you detect in the ones that survive. Is there a way, it's hard, I know, but is there a way to like purify those cells before they die, so you can clean up your surviving transcriptome from those targets that just simply rise because the CTLs are trying to kill the cell?
Yeah, it's a really good question. It's one that our bioinformaticians raised at the beginning of these experiments. We're looking at what is kind of subtracted out, but we're missing that comparator group of cells that recognize CTL and went on to die. So your question is there a way to kind of capture them before before they die and see what
Exactly.
functional changes look like. I haven't thought about it in a while. I'm not sure there's a way to capture them in the process of these experiments, but the type of thing we might be able to do would be to knock out some of the death mechanisms like perforin and cytotoxic T cells, and use those kind of declawed TTLs to see how those are perturbing the target cells. It might not be exactly the same because they're not, they're not responding to the impact of being perforated, but we'd probably still see that interferon signature that we're speculating comes from these close encounters. So, yeah, thanks for raising that as a question. I think we can think about that a bit more. Maybe there are some things we can do.
Thank you.
Are there additional? Melanie, you can go ahead.
Yeah, very quick question. Great talk, Brad. Can you speculate a little bit more, talk a little bit more about the poliovirus receptor and how you think that this is, this receptor is involved sort of as a checkpoint or, NK target, or how do you see that?
Yeah, so the way that we've envisioned it, even though we don't have functional data on it yet, and there also wasn't functional data in Matthias's paper, but the way that I think we're both envisioning it is that this ligand on the target cell will engage with TIGIT, which is co-inhibitory receptor on the CD8 T cells or the natural killer cells, and in that way impair the function of those effectors in a way that protects that target cell. That's the idea. But kind of perturbing that system is something that clearly kind of needs to be done on a next step, and ideally done against real ex vivo reservoirs from some of these folks who have overexpression of PVR in their reservoir cells.
Is there any indication that this receptor is overexpressed on latently infected cells?
Yeah, so that was actually in Matthias Lichterfeld's recent Nature paper. They have a way of, I don't know the details exactly, but they have probes that can detect infected CD4 cells without reactivating them, so unperturbed cells. And there's enough of those probes that they can distinguish intact from defective proviruses. And then they have a site-seq-like method that allows some phenotyping of those cells. And what they found as the marker that was most differentially expressed in reservoir cells with intact proviruses was overexpression of PVR.
Thank you.
Are there any other questions? Okay, well, thank you very much for a great talk, Dr. Jones.
Thank you. Thank you all for the questions. Appreciate it. Attention.